Follow us on social media and please hit the subscribe button so you don't miss out on any updates from our ALS experts.
All right, folks. Welcome to another gathering of your extended family. The people that you find here are your friends, so don't be bashful and ask any questions that you could possibly come up with. Nothing is too little, and they're all important. And I would just like to share a little something with you all. You know, each one of your stories is amazing, intricate, and we appreciate that you share your stories with us. Well, I have a story, and in the chat tonight, there's going to be a link, and I have given my audiobook for free to all of you. It is on a YouTube channel, and there is a link in the chat that you can copy, and in a short while, it will be on our website, and you will be able to hear me express what happened to me 16 years ago. And I would really appreciate if each of you could share that with somebody else. And with that, I just want to say thank you for sharing yourself with all of us. And over to you, Lisa.
Thank you, McFinn. So welcome, everyone. I'm Lisa Deegan, for those who don't know me, and I'm the co-founder of Everything ALS. And I want to thank you all for joining us for tonight's ALS Talk Series. And we're so honored to have our guest speaker, Dr. Jonathan Katz from CPMC and Cedars Medical Center. He's going to be presenting to us tonight on thoughts on clinical trials from the perspective of an ALS Center director. So just a few quick house items before we get kicked off. I want to make just a quick announcement. Then we're going to have Dr. Katz present. We're going to follow up by Q&A, and then we do our non-recorded open forum portion. So Everything ALS likes to empower people through education in the latest research and treatments, clinical trials, and updates in ALS from the top experts around the globe. So we're so proud to be providing people with access to the experts and get your questions addressed. We offer a safe place for people to meet and have open discussions about what is ever on your mind. Clinical trials are more successful when biomarkers are involved. So through our citizen-driven research, we are working on discovering genetic biomarkers along with digital biomarkers, as they are important in ALS to help us understand the cause, prognosis, and to help diagnose people much earlier. Digitizing the gold standard ALSFRS scores so we can better measure in clinical trials will help to accelerate therapies. Our speech study focuses on diversity and inclusion, and it's accessible to all. You just need to be 18 and over living in the US. It is the largest voice study in ALS with over 1,000 hours of data collected. We have a participant portal where you can access your data and share it with your doctor. And we have the best of the best experts collaborating and analyzing this data. It's only 10 minutes a week. You can help to save thousands of lives now and in the near future. And collaboration really is key on this. We need to work together as a community. So please visit everythingals.org/research. And so now for tonight's presenter, we have Dr. Jonathan Katz. He is a top neurologist in San Francisco and is affiliated with multiple hospitals in the area, including Sutter Medical Center and California Pacific Medical Center. Dr. Katz has been in practice for more than 20 years, and his work in ALS has included leading clinical trials and pharmacotherapies to slow progression of ALS, device trials, diaphragm pacing systems to support weakness, and observational studies of factors affecting disease progression. In collaboration with his colleagues at Forbes Norris Center and UCSD, he studied the mutations in RNA binding proteins that cause neural dysfunction and death, and collaborations on new stem cell and gene therapy techniques to treat patients with Cedars-Sinai Regenerative Medicine Institute. So we actually did have a webinar on that with Dr. Clive Svensson. So Dr. Katz is president of the ALS Research Group, an international ALS research collaboration. And he sits on the medical advisory board for GBS-CIDP Foundation International and Myasthenia Gravis Association. So please join me in welcoming Dr. Katz. Thank you for joining us tonight to share your insights and your expertise on clinical trials in ALS.
Can you guys all hear me?
Yep.
Yes, we can.
And you can see the screen, right?
Yes.
Yes.
There's nothing like sitting in your office by yourself, staring at a computer and talking to hundreds of people. I was flipping through the slides, and I probably know about 25 or 30 of you. So I'm happy to see some of you on the computer, and some of you I haven't seen for a long time. You know who you are. I'm happy to see you're doing okay. And for those of you I don't know, I'm happy to sort of meet you and not see your faces in this great pandemic that is keeping us apart. Anyway, I thought I would just give you guys an honest assessment of what it's like running trials at a clinical center. It's not a talk that I've given. Hopefully I'll get it done quick enough so you could ask a lot of questions, but as you heard my credentials before we started, I was before I did ALS, I was working in other diseases, and I started working in ALS in about 2005 with Dr. Bob Miller at CPMC. And some of those organizations you've heard about don't even exist anymore. And some of those associations that you heard about I don't really have anymore. But over the years, I've gotten more and more involved in ALS clinical trials. And, you know, the clinical trialist part of ALS, if you think about it, is just one piece of a lot of different pieces in this field. One of the pieces is you guys, it's the patient support group. There's also the associations, the patient support groups. There's the scientists who study the disease. There's the companies that are involved in these trials. And, you know, somewhere at the center of this, you have people running ALS clinics. And at those clinics, you have some people doing trials. And we're really just talking about the perspective of that one small area of what is really a collaboration between a lot of groups. But I'm going to try to just be as bluntly honest as I can about some of the things we think about and some of the history of this and some of the roads we've been on. And, you know, some of it, I'm not going to be like super promotional of anything or, you know, say anything that is a little too cheerleadery for the sake of saying it. It's been a really long road. That's what this first slide is about. And I wish more than anything, you know, that I could look a patient in the eye and say, we got a cure for this disease, and I could either make it stop or even better, make it get better. But the reality is that path from A to B is pretty long. And, you know, sometimes it feels like it's a lot longer than you want it to be. I thought when I joined this field 15, 17, 16 years ago that we'd be seeing a full cure by now, maybe, but it's really a very challenging area. And, you know, it leads to certain kinds of trial discussions about trials and the reality of trials and some of the tribulations we go to. And, you know, right around the time I was entering this field, people had just gotten done doing trials on Celebrex and gabapentin. And, you know, these drugs, Celebrex and gabapentin, it's kind of like you look back at it and you're like, what were people even thinking? It's like kind of like, you know, it's kind of like the first handheld devices, those big thick things that you were talking on the phone about. It was a completely different era. And it kind of speaks out to the question of which the answer is true. People are sort of guessing at it at the beginning. And it makes you ask the question too, how far have we really come along since then? And, you know, we hear from all the scientists that we can attack all these various pathways of the disease that are listed in this slide. But, you know, to a clinician, what I don't hear or don't yet feel that there's an answer to is somebody just sitting down and saying, here's exactly what happens in this disease. Here's how it starts. Here's how it progresses. Here's why it progresses from one area to another. And here's what we're going to do to make it stop progressing. So we kind of have these pathways that are almost separated from each other in time and space. And the scientific approach is to look at one pathway or another, but we really need it. What I think we really need at some level is sort of like an all-encompassing theory of the disease that we could hang all these ideas on. And that's sort of what I, as a researcher, look forward to hear one day, you know, hopefully from some of the better scientists in the field, a story about, like, you know, here's what happens to the brain, and here's what forms in a cell, and here's how things evolve from that. And I think that's going to be essential. Sort of a grand encompassing theory of ALS is going to be essential to hang some of these individual ideas on to really get a sense for what causes the disease. Now, one of the things that I do find interesting is some of this genetic stuff that's going on right now, but that's for a very small percentage of the patients, where you could actually block the defect of the disease. But even with that, which sounds so optimistic that you could just turn off a protein that's toxic to the disease, to a degree, we don't really know 100%, particularly for C9. Is the aberration in the protein something that's triggering the disease to start, and then you just get regular ALS and we treat it the same way? Or is the nature of the progression of the disease from the protein itself, and by getting rid of the protein, we could stop the progression? It actually seems to me more like it's going to turn out to be the former, that this mutation is just a trigger for the disease. And then, you know, people like to ask the question, like, why has there been a lack of success for ALS? And, you know, these are just some things I've jotted down over the years. Why has it been so slow to get from point A to point B? Some of it is that, you know, we have the wrong animal model. The SOD disease that the animal model is mostly from is different than the ALS, where you have TDP-43 as the underlying pathology. So maybe it's the wrong animal model. We don't have a great high throughput model yet, so there's not a way to say, let's just test drugs and see if they affect the disease. There's a lot of ideas in that direction, but we're really not there yet. And then if you go on and on, people like to point a little bit towards maybe our outcome measures are not that good. Maybe our studies aren't big enough. There's not enough funds. I think the truth of the matter is we don't understand the disease well enough at this point. And, you know, we're trying hard. And I think that should be a really big emphasis of what any group does for this disease. And in the back of my mind is just like, step one is understand this thing a lot better. Now, for somebody who's doing clinical trials, there's another question that sort of springs from this, and I'm going to start talking about it as we go forward. But just from the science of the disease, I think at the core, the thing that has to happen is we just have to understand the disease a lot better and how these different pieces and pathways come together. Now, one of the things that I've been involved with is just asking the question, what makes a good drug to study? We do have all these pathways. We have some understanding of the disease. Optimistically, it's a heck of a lot better than it was when I started 15 years ago. And anybody who's paying attention to this disease can see that we're studying more and more drugs as time goes by. But, you know, I put up this Magritte painting on the right in this slide, and I think that's French. I don't speak French, but I think it says, "This is not a pipe." And the joke there that Magritte was playing on his audience is that it's not a pipe, it's a painting. And that's sort of something that reminds me a little bit about ALS, in the sense of we don't have an all-encompassing model of the disease. And what we're treating is ideas about the disease still. And when you try to figure out like what makes a good drug, we could point to all these different pathways and we don't know which one is the one. So we're not really treating ALS, we're treating like individual pathways of the disease at this time. So how does somebody who's doing trials proceed in that environment becomes a question. So the first question is, what drugs are you going to study as a trialist? And I think at the core, a lot of it is like, does the person who has the drug, the company that has the drug, have the wherewithal to finance it and put everything together? I completely admire anybody who could do that and get the excitement together. I completely admire the platform trial, the companies that come into the field that can do the business to make these studies happen. It's really important, given where we're at, to study drugs. And from a patient perspective, the patients who see me, for Lord knows what reason, people get much more excited about some trials than the other. People got very excited about the stem cell study, which we did at our center. People sometimes get less excited about other theories. I'd love to hear from some of you what makes for a good drug in your mind as you go forward and study. And I think one of the things that sometimes is helpful is that there's some evidence that it's already been affected in a small trial of humans or in some trial of humans. We see that over and over again. But I even wonder sometimes if, when I'm talking to patients, it's more like door number 3 on Let's Make a Deal. Like, you might see a drug that's in a trial that we know from an earlier study is 25% effective, but maybe you don't want to enter that trial because you want to go for a drug that's 100% effective. Hence, what's behind door number 3 might be more attractive. That drug that's 25% effective, it would be good if it's just on the market and people can use it. But it's sort of like we're going to try to keep hopping. And if you're going to try to keep hopping to the next drug and the next drug and the next drug, you kind of have to start thinking like, what strategy should we take as a field to study drugs? How many drugs should we study? When should we go further with a drug versus giving up on a drug? So all these things come into play. And then I put a couple of slides in there about just who gets into a trial, because I know that's a question for patients too. So you're sitting at the bedside, we're moving past which drugs you study now, and you're sitting at the bedside and you're trying to explain who can be in a trial. And I think it's important for people to hear, like, the big thing you're thinking about when you're doing a clinical trial is you want to get patients in the trial that are likely to change in the time that that trial is occurring. So as a clinician, when you're looking at a patient, there's certain clinical presentations that are different than others. Here's just a picture of somebody who gets very weak in their arms, but nowhere else in their body. And it lasts for months and months, and then years and years, and nothing seems to happen, but the arms get weak. Somebody like that's going to be very bad to put in a clinical trial because they're going to have 0 points in their arms and they're going to have 4 points everywhere else. And if you put them in a trial for 6 months, the odds are pretty good that's not going to change. So somebody like that, sure, you want to help them in every way possible, but for a trial, it's just the wrong kind of patient because those points are not going to change very much over 6 months that you have in your FRS score, and it's not going to be good. People with primary muscular atrophy don't have brisk reflexes, on the other hand. For some reason, we don't allow those patients in the trial, in trials. I think that could possibly be a mistake. There's not a lot of patients who get excluded for not having any changes in the reflexes, but just people are thinking about this and we are evolving as we go forward. I put in here another clinical presentation you sometimes see, which are people who the predominant feature at the beginning is breathing problems. Those patients often can't be in clinical trials because their breathing is such that you worry that in 6 months of a trial, they might not survive over the course of those 6 months. So if you put them in a trial, you're going to be missing important data, which is how much progression there is. So generally, we want people to have good breathing when they come into clinical trials. So these are just things that we are thinking about all the time. And then frontotemporal dementia in 10% of ALS patients. Well, we'd love to put those patients in trials, but the problem is the dementia can make doing some of the measurements, in other words, getting good answers to questions like about your function, make those things very difficult. So we don't necessarily put those patients in trials. And it's also, for some reason, those patients with FTD sometimes tend to not live as long as others. So there's reasons we're thinking about these things. A lot of it is just about numbers. And when you're talking about numbers in our trial, you're talking about the ALSFRS score. And the ALSFRS score is, I'm sure you all know this, but it's a score that is 3 questions each about your mouth or your bulbar region, your hands, your fine motor skills, your walking or your turning, gross motor skills, and your breathing. Each one's worth 4 points, 12 total questions, 12 times 4 is 48. And people start, as they get weaker, the score starts falling from 48. Usually at the time people progress, scores around 40, present, the scores around 40 or 41. Usually by the time trials start, the scores around 38, 39, and you go on and up from there. But that score, it's important to realize when you put patients in trial, if you just think about numbers, you don't necessarily do very well if people have a lot of 4s in those numbers, because 4s mean there's nothing going on. So if somebody comes into a trial and there's 4s there, if they don't start to progress in that region during the 6 months that they're in the trial, the scale's not going to work very well for that person. So if their hands are completely normal, the scale's not going to work very well. If the hands are already getting a little weak, if they've gone from 4s to 3s, there's a good chance that they're going to do okay in the trial. So every region, in theory, should be dropping some points. And I'll say that the respiratory region, because we don't want people to get sick, we tend to say that region cannot be affected when you start a clinical trial because you run too much risk of getting into problems breathing. Well, that essentially knocks 12 points off your trial. So you're working on the remaining 36. So you just start getting a feel for how this scale works. The other thing you have to be careful of when you choose who goes into a trial is that you don't want a lot of 0s and 1s on that scale either. So you have 0s and 1s, what problem do you run into? You have what's called floor effect. You can't decline anymore. So that part of the scale also becomes useless. So you're really looking for like a Goldilocks sweet spot when you use this type of scale. And it might sound, cruel that it makes some people not able to enroll in trials, or it would make a trial less perfect if you put people who have a lot of 0s and 4s in the scale already. But when you look at the trial that Mitsubishi did with Radhikava a few years ago, they basically created criteria that more or less ensured that people would be at 3s and 2s on this scale. So that's a good way to use the FRS, and we began to learn from that how doing relatively small trials could be helpful. The other thing we're thinking about when we do clinical trials is that ALS is a very heterogeneous disease, right? Like, as you all know, I showed you some different clinical presentations at the beginning, but people can have the same presentation and one person progresses very fast and one person progresses very slow. So you're really, again, you're looking for a Goldilocks phenomenon. And the way we do that is, we tend to know who's going to progress at what rate. We have information about what sort of things lead to better or worse prognosis. So like limb onset is better than bulbar onset. And for various reasons in our trial criteria, bulbar patients have a harder time getting in than limb patients, because when you have bulbar involvement, your mouth doesn't work. So it's hard to do an FBC because it's hard to get a seal. So that number tends to be lower. And like I said earlier, there's FBC cutoffs because of respiration. So what happens is we trend towards having limb patients in the trial because it's harder for bulbar patients to get in. Also, patients who've taken a very long time to diagnosis or a very high function at the time of diagnosis tend to do better than patients who are diagnosed very quickly. And that's just because the disease is progressing faster. When you run a clinical trial, you have to be careful that you're picking an amount of time since the disease started for your cutoff. So some trials say you have to have disease within 2 years. Some trials say you have to have had disease within 3 years since it started. If you think about that, what happens if you allow people in the trial who've been progressing for 3 years versus 2? Well, by the very nature, if your breathing is still good at near 3 years, you have slow progression. Everybody can see that, right? It's not progressing very fast. If at 3 years you're breathing and talking perfectly, so you could get into a trial. That would mean that, some trials are afraid to put people in who are progressing too slow for the same reasons I told you earlier. If the progression is too slow, You're not going to see anything in 6 months. It's hard to see change if a lot of your patients aren't progressing, and you don't want to run trials out to 12 months or 18 months. So the slower progressors after a period of time get eliminated by these time cutoffs. And then LS-Goriel criteria, the people who have a lot of involvement have definite ALS. So if your arms, legs, bulbar region is involved all together, let's say that's definite ALS for the sake of simplicity. Patients like that are, in a sense, the best for trials because all their points are dropping at the same time, if you think about it. Patients with very focal disease, like I showed you earlier, where it's just in their arms, they could be problematic in trials because their scores might be 0 in the arms and forearm. So these are the kind of things we're thinking about. And the picture on the right is just to say, in a sense, we're excluding the extremes and looking for those patients that have conditions that are right in the middle. And the other thing is, like, I took this from a company's website and I just put the funny line at the bottom that says, this can't possibly be correct. And this is something that I'm just like going to say to you guys, because a lot of you are patients and I want you to understand it. A lot of stuff that goes onto the internet or goes up in the world, it has good intentions, but I think at the core, at the end of the day, you have to do a placebo-controlled trial at this point in time to understand if a drug works. And this is something I just picked up off the internet where it says the ALSFRS, the scale I showed you 2 slides ago, is a simple, reliable tool for evaluating the decline in function. Scores decline with disease progression at a rate that is generally consistent across clinical trials. And I wrote that can't possibly be true because different trials are enrolling different patients. And I will tell you that if I create a trial that I allow 3 years since the disease started versus a trial where I allow only 2 years since the disease started, the progression is going to be slower in the 3-year trial because I'm letting more slow progressors in. People have to think about this because you only have so much money for a trial. You can only enroll so many patients. And if you include everybody, you're going to get more slow progressors if you go through. So this thing about 1 point drop, when you look at something like that on the internet, I think the meta message there is they're trying to say to you that they have some data that looks better than a 1 point drop. But any data that you see that doesn't have a comparator to it is very hard to interpret in a disease this heterogeneous. So I think that's important to remember. So again, that overall outcome of a trial, that whether the average drop in a trial is going to be 1 point or 0.7 points a month, or if it's going to be 1.3 points a month, it depends on what kind of patients get enrolled in trials. The perfect clinical trial is going to have all patients sort of in the same zone as each other. So there's not a lot of heterogeneity, and it's easy to measure that group against the control group. This is kind of what we're getting at when we're doing the trial. So now I'm sitting with a patient who comes into my office. They might have new ALS, or they might have just come to ask me whether they should do a trial. And I have to explain to them, like, what to expect, why should you join the trial? And the things that we generally talk about is like what I just said. I hope I convinced many of you. I'm going to talk for about another 5 minutes, and then we can ask questions. But placebo trials, unfortunately, I think they're necessary to understand what's happening with the drug. The flip side to that is you don't go into a trial, you're not getting the treatment for sure. So you're actually, in a positive sense, increasing your chance of getting a drug that's helpful. Now, I think the way the field's going, and you all probably know this, is the treatments are probably for now, until we get the perfect treatment, going to build on each other. So Riluzole was the first treatment, Radhikava the second treatment, hopefully Amylex, the third treatment that looks like it might be effective. And all of these things are going to sort of, in my opinion, summate to slow down the progression of the disease. And, Riluzole was 10%, and Radhikava, the estimate on it's 33%, and Amylex, the estimate on it right now is 24%. You start adding that up on top of each other, you go from 100 to 110 to 33% plus of 110, which is about 145, to 20% above 145. You're starting to get to slow this disease down a lot. And I think that's optimistic. And if people can say to themselves, if I get on those 2 drugs and maybe eventually a third one, and hopefully get a trial drug on top of it, we really are hopefully going to get into being able to slow this disease down a lot by affecting a lot of these different pathways. And fortunately, clinical trials are easier, at least at our center, they're relatively easy to do. I think patients want drugs and they're willing to take risks. And the clinical trialists and the people who run the trials are starting to get the message of providing open label extensions and expanded access programs. So you get that benefit if you join a trial. And right now, I would think we're actually getting into a phase where there's more drugs that we need to study than there are patients. So we use this thing called the 5R approach, and some of it's a little sarcastic, but I think it gets the point across. The 3 drugs we have now are riluzole, Radhikava, and a research drug. Unfortunately, the Amwits group, I don't think, is going to pick a drug that starts with R. I put reality as the fourth one in there, which is really what this discussion is all about. What can we really do for you at this point in time? I get a ton of questions about things that we just don't know the answer to because they haven't been studied in trials. And that's sort of what reality is about. And retirement is about, when you have a disease like this, living happy, which I think is a super important part of everything. And then just a couple of comments. I know somebody's going to ask me about Radhikava at the end of the study. Radhikava had 2 trials, and I think actually, in my opinion, what they did was sort of brilliant with respect to the way they used the FRS. They did one trial that showed a hint that the drug was effective, and they thought the trial wasn't effective because they had too many slow progressors. And if you have too many slow progressors in a trial, you will not be able to see a result. So they changed the criteria for their trial, and they did a second trial, and it showed a decline with a really good statistical significance, 33% between the placebo group and the treatment group. Now, these are the criteria that they used, and those criteria are pretty restrictive. You're damned if you do, if you're damned if you don't. The insurance companies didn't just jump in and give it to everybody, which is what the FDA wants to do. But I think all in all, the way that the Japanese used this trial and manipulated this scale to get patients in and prove a drug was important was actually pretty brilliant, and I'm pretty positive on that trial. I know other people aren't as positive. A lot happened when that drug hit the market in the first place. I use this as a joke because the drug was studied in Japan, and it got pushed onto our shores, and it almost felt a little bit to some people like Pearl Harbor got attacked the second time. I think when the drug first got used, it was expensive. I don't know what I meant by patients don't want wine. I think I just typed something and was thinking about something else, but I figured it out. But it was difficult to get approved by insurance companies. Our centers had like 1 million patients coming in at once. And, there was a question about uncertain benefit for some reason, but I think a lot of this stuff just sort of like crept up through the ethers. But if you really look at that trial by itself, it's pretty interesting. Now, the other thing that I think happens is like 10 years ago, we did a small study on a drug called dexpramprexol. It was roughly the size of a lot of these smaller trials that are going on right now. It wasn't the greatest small trial, and I won't tell you what the details of it were, but it wasn't as good a trial as it could be. It was followed up by a gigantic trial with 943 patients. The trials that are being done now have about 120, 130 patients in them. And I don't think we're ever going to see anything this big again, nor do I think we have to. I think people have actually, from a trialist standpoint, gotten a lot more clever with how to do clinical trials. But there's this lingering thing because you went small trial, not approved or even considered to be approved by the FDA, to much, much bigger trial, it didn't show effect. So I think that led to a little of the subsequent skepticism. I will say that the small-ish trial of Radhikava was way, way better than anything compared to the preliminary study of dexpramprexol. And then, as I said earlier, Amylyx is coming. They have a really nice study that got done with good criteria. The study was relatively small, so we'll see what the FDA does. I'm free to discuss. And then the last story I want to tell before I open up to questions is the Brainstorm study that just got done, which also, just as a rough number, had about 200 patients in it. It was motivated in part because there had been some earlier work suggesting patients might have done pretty well on the study. The trial design itself had some flaws in it, and I won't go into it in great detail. And I don't blame anybody for the flaws because you had to make stem cells, and that took some time. And because of the trials that had gone on earlier, they were trying to find patients that progressed pretty quickly. Those two things, taking the time to find relatively fast progressors and then making their stem cells, pushed the starting time that patients began that trial back by several months. And because of that, their starting FRS scores were the lowest of just about any trial that had ever been done. They were a 31, where usually trials are starting where the FRS score is about 38. So what happens? What did I say at the beginning of this talk? To go from 38 to 31, you're going to get a lot more floor effect, meaning there's going to be too many patients starting with numbers like 0 and 1. And in fact, that's what happened. And it kind of made it difficult to see the result in the trial. And what they were able to do is show in a relatively small group of patients, but you could still measure the results because it didn't get too close to 0. In other words, the ones that started without that much weakness, there was a good hypothesis to be made that Neuron was doing okay in that trial. So it brings me to sort of my last statement about some of this stuff, which gets to be the platform trial that's going on right now. Platform trial, which is brilliant in my mind, is so you can study a lot of drugs at once. And the strategy of ALS drug development should be to study a lot of drugs and throw out the drugs that don't work really, really quickly. Because you want to be able, since we don't know which drug is going to work, and since we learn something when we do see an effect of a drug about pathways that might be important, I think you want a lot of patients in trials and you want to study a lot of drugs as quickly as possible. The flip side of that is when a drug does show that it might have some efficacy, I think you need to keep that drug in play and do the second trial so you could get a closer look. And I would say some of the trials we've seen recently have not worked, but some of the trials I just showed you, there's hints that there's at least some efficacy, and I think it's important to follow up on that kind of strategy. The very last thing I want to say, I promised myself I'd be done by 4:40, but I started 10 minutes late. So this is when I was like, 10 years ago when I started, right to try was a big thing. And right to try makes a lot of sense, right? Like people should get access to drugs. But for me, the reality of right to try at the very beginning was that it makes sense, but it's not realistic because these drugs aren't there. And I was worried when the thing first started that if this thing is not done right, it's like American politics. On the right, you have people who support corporations. So they're going to want those corporations to be able to sell whatever they want. Sort of what, some of our politicians call libertarianism. On the other side, you have sort of the left who believes anybody should get anything that they want and the taxpayers should pay for it. And those two things together, I think, create a bad dynamic because if you really don't think it through, you run the potential of pushing drugs out that aren't effective and people having to pay for it, or people it creates a potentially bad dynamic, or making the taxpayer pay for it. So I do think we have to be much more deliberate than just saying there's a right to try, and not to mention that who should go into a trial to find out if a drug's effective when you need placebo groups. So to a trialist, this is a really hard question. Now, obviously, a lot of you are behind this next thing, which is the Accelerating Access Act, which is wonderful that so much money is coming into our field. But I will tell you, the clinic directors are rightfully concerned about this act for a lot of different reasons. Some of it, what I just told you about right to try concepts, taking drugs that might not work, as long as there's checks on that, like take the drug up until we get the result of a trial, it makes sense. But also because maybe it wasn't fully thought out that some of the clinics that run trials are going to have a hard time without infrastructure developing, just being able to get these theories to these drugs to people. So we kind of want to see that the right infrastructure is out there for these critical therapies to work, and that the clinical trials is not forgotten in the sort of financial metric that accompanies this. So I'm here to answer questions. I'm sorry I was late at the beginning, so I will stay happily 15, 20 minutes late if you guys want. So thank you. And now my worst moment of all is worrying that nobody heard one word I said for the last 45 minutes.
You don't have to worry about that. We
Okay.
heard it, and we've got quite a few questions to ask you. My name is James. I'll be one of the moderators asking the questions. I'll start off with, has there been any long-term studies on longevity comparing someone who survives, say, 10 to 20 years compared to people who do not survive for more than 2 to 3?
I mean, you know, the answer to that question is super important, and there have been studies on that sort of thing, of course, but there's not an answer to that kind of thing yet. So it's going to be really important. Like, everybody wants to know at a cellular level, what is the difference between somebody who lives a long time? Are they two different diseases, or is there a difference in whatever transmits the disease from one cell to another in the person's body, where maybe the same pathology just goes slower in one person than another? There's secondary genetic issues that sort of link on to the pathology that speed it up or slow it down. Incredibly important question. But I think the answer to that question, like the answer to many of the questions, is going to come once we understand the overall theory of disease, because that question is just the flip side of how does the disease start and progress. And everything has to hang on that grand unifying theory, and I don't think we have it yet.
Awesome, thank you. And I just wanted to introduce myself before I jumped in. My name is Zoe. My father was diagnosed with ALS in 2017. So I just wanted to say before I get into the questions that this was a really, really great summary of all the trials and stuff. And I'm a neuroscience major, so this was really cool to kind of like talk about the mechanism and like the biophysiology of the disease and how much we know. Anyways, so the first question, or the second question is, are the trial designs that study slow progressors, are there trial designs to see how slow progressors have reached that sort of longevity?
Yeah, so you tricked me a little bit there, Zoe, because I'm not 100% sure that was a different question than the first question. But so are you asking, are there studies that show how slow progressors are different than fast progressors, or did I not understand it?
Yeah, like if, oh, I guess two people have asked the similar question. Okay, yeah, I guess you kind of answered that already. We can just move on to the next one then. The next one was, does the ALSFRS score equal staging info?
Well, staging is sort of different. Like the ALSFRS score, if you think about those letters, functional rating scale, right? So it's exactly what those questions are. They're all questions about somebody's function. So how well do you use your hands? How well do you swallow? How well do you breathe when you're lying down? Like those are just function. And we can score them 4 points each by, breaking them up into I breathe great, I breathe less great, I breathe not so great, and so on and so forth. Now, staging is sort of a different question, and staging to me can be a little artificial in the sense of like, what is a stage of ALS? Like I can say you're in an early stage, a middle stage, or a late stage. And there's been people who develop staging mechanisms, staging algorithms of the disease, but I'm just not sure it helps that much because let's say somebody starts with a breathing problem, and it's a bad breathing problem. In fact, I see patients every once in a while. I saw one this week who presents with ALS in an intensive care unit because the only thing that got affected was breathing. Nobody ever noticed, or the doctors weren't paying, we couldn't figure it out until it was in the intensive care unit. And, that patient obviously is in a late stage of disease, but only one part of their body is involved. Somebody else could get ALS just in one arm. And it's the same progression. It's just the nerves in the arm are progressing. So staging, it doesn't mean, in this disease, it doesn't mean that much from a research perspective, or even in a sense from a clinical perspective. Now, if you just took all the patients who have sort of involvement everywhere, and you said, like, you're in an early stage at the beginning and you're in a late stage at the end, and tried to break that up into pieces at different points, you could probably come up with some kind of scale, but it's just not as helpful to talk about stages as it is to talk about function, in my opinion.
Makes sense. And the next question, I think, is actually a very interesting one. Why can't placebo data from all trials be crossed and made into one average progression rather than having to repeat that placebo at each different trial?
Oh boy, that's the $1 million question. And it's unfortunately going to take me from 4:59 to 5:59 to answer it. So I'll try to give you it's dangerous to do that. It makes all the sense in the world to say it's probably important to understand the answer to that question better. You can't just take a trial placebo and compare it to a different trial's placebo arm or to overall placebo arms very easily, because even if I use the exact same criteria, I said, give me all patients who have FRS scores below 32, I'm completely making stuff up, and I compare it from the trial I'm currently doing to an old trial. Even if I use the same criteria, I might get slightly different patients in my current trial because the older trials that had placebo groups were just enrolling slightly different patients. Because, well, let's say I'm doing a trial that cuts patients off at 3 years, and I compare it to another trial's placebo group that also cut patients off at 3 years. But it just so happened that I'm doing my trial with the exact same criteria as that trial. I'm saying I want to use that trial as placebo arm. But it turns out that when I'm doing my trial, something sneaky happens, which is somebody else does a trial that has a 2-year cutoff with a slightly better drug for people to want to try. So all of a sudden, even though I cut my trial off at 3 years, I get no patients in that trial from 0 to 2 years, while the other guy who's running the same trial does. So you get totally different outcomes in that placebo group because of the nuances of what's going on at the same time. You're really taking a lot of risk if you control a trial with the placebo. Now, I think what should happen and what the field should get much, much better at is taking a database and taking some really good statisticians and being able to predict the outcome of every single person that goes into a trial. Because when you sum up all those outcomes of all the people in a trial, I think you should be able to get a really good estimate of what the outcome is. So if, like, I'm enrolling somebody in a trial who, has an FRS score of 48 and onset 6 months ago, or say 43 and an onset 6 months ago, and has just arm involvement and a foot drop, I should be able to find other patients that have sort of the same prognostic criteria. And across a relatively large trial, I should be able to predict pretty well what that group's going to turn out. I really do believe that that's true, but I've not seen anybody do that yet. And I'm kind of, I understand math pretty well as a doctor, but I don't understand math that well as a statistician. But I would really like to see somebody using one of these prognostic criteria to put them against every single patient in the trial and predict what the outcome of the trial cohort is going to be. based on, say, the predictive criteria of about 100 different patients and see how close they could come. If I were a patient support group, I'd put out a prize for somebody to do that accurately. And then maybe that could lead to say, hey, here's the patients I enrolled in my group. The outcome of this group should be, an FRS of 1.3 over the next 6 months. I really believe that sort of math is doable, but I don't know why there's not like 5,000 papers on this already. So it's just my hint to you guys.
Wonderful. And hopefully we will be moving in towards that direction. Okay, so the question is actually something that you predicted that they were going to ask about, which is, are you personally a supporter of Radhikava?
Yeah, that was in my talk, and I kind of made it obvious. Yeah, I mean, here's my thinking about Radhikava. It's, in my honest, humble view, and I'm not saying this, I mean, I have lectured for the company, but it's not why I say this. It's really just the way I think. And for those of you who believe in conflict of interest, it's like maybe sometimes the conflict of interest goes in the other direction, where you're speaking because it's coming from your heart. But I think unless we were completely like misled through fake news, which, these days you got to worry about, but assuming, and I have no reason not to assume this, but assuming everything in that trial is on the up and up, which I'm sure it is, that's pretty good data. And it's the best data we've ever seen in this field. Now, I get that people have to right now get an IV line under their collarbone, but the truth of the matter is that's the best looking data we have in this field so far. So if somebody comes to me and says, my goal is to take the best drug that we know about in this field right now, that's the drug. And it doesn't make a lot of sense to me to this day why everybody sort of turned negative on it. Like, that is a trial. Somebody's going to have to explain to me, or I'll put it the other way, what's wrong with that trial? What don't you like about it to get me to change my mind? So I don't understand some of the negativeness about this stuff. I particularly don't understand negativeness from, doctors that think we should be prescribing, supplemental therapies for the treatment. So to me, I'm looking at the best clinical trial, well-designed, smart protocol, everything's logical, and you get this good result. We don't have anything better. You can tell me that there's reasons it might not be right, but we definitely don't have anything better. And that's a pretty good scientific project by the people who did that study. And I think they should be congratulated.
The next question I have is about neurons specifically. I'm sure you're very well versed in it. The two-part question first off is, could you perhaps elaborate on neuron and the reversal of symptoms in some patients? And as you spoke earlier, with ALS not fully understood, why do you think some drug trials like neuron have shown success rates?
Well, don't get me wrong here. I'm not saying ALS is not understood. It's just understood in parts, right? So you don't have a grand unifying theory yet. It says, here's, like I said earlier, here's how the disease starts. Here's how it gets from cell to cell. Here's how it gets from part of the brain to part of the brain, from nerve to nerve, like all the pathology that goes behind that. So, with other diseases, when you're a medical student or a neurology trainee, somebody sits down and says, here's the antibody blocks the muscle doesn't work, get rid of the antibody, the disease gets better. Okay, that's really easy. You can't do that in ALS. Like, tell me what's going on. You don't know what the answer to the question is. So, but people say, we can look at all these different things that are wrong with cells with ALS and try to reverse some of those things. They just don't, when I say they don't hang on a grand theory, like somebody could tell me there's a problem with mitochondria, but where does that fit into how this disease starts and progresses? Somebody could tell me that there's funny inclusions in cells. Well, explain, how do they form? When do they form? How do they affect the cell? How do they move from one cell to another? That's what I'm talking about with the grand unifying theory. That does not mean you can't do research and try to use the research to cure patients, which is the most important thing. But the second most important thing is to begin to say like, oh, that treatment has some input into this, because that helps you put the pieces together. Like it's deductive reasoning until you finally go, aha, there's your theory. It makes sense. So it's all important. It's all like that slow arc towards success. And Neuron, I think, had some really good theories behind it. And, you know, they wanted to study the drug. I don't think patients should hang on the idea that it's going to reverse your symptoms. I think we should look at neuron like we look at any other treatment, with the goal right now of slowing down the progression of the disease. We're not there yet. I wish we were, but we're not there yet to say we're going to stop the disease or reverse the disease. That, I think, is way too optimistic from reality. I do think that theory ended up in some of the DNA on how that trial got designed in the first place. But when you really look at the data of that trial, there's some optimistic data in there that hopefully will be followed up on.
Wonderful. So the next question that we got was, how do other treatments that you may be taking as well as a trial treatment, how does that interact with trials, or how does that affect someone getting into a trial?
So in general, the two treatments that, you know, are on the market, Riluzole and Radhikava, you have to be on a stable dose. And different trials designed to find stable doses different ways. Some trials define it as, you know, as 14 days on the drug, 30 days on the drug, whatever. But you have to be on a stable dose or just not take it, because what trials generally don't want is people starting drugs in the middle. And then the other thing, trials tend to exclude quote unquote experimental drugs. And obviously the idea with that is if one of those experimental drugs either causes bad side effects or actually works or doesn't work or actually can make a patient worse, it's going to start throwing kinks into your trial. So experimental drugs are generally excluded in ALS clinical trials, just because it makes everything too confusing. And, you know, I would throw in there, I think that's the right decision, because, you know, when you really think it out, like experimental drugs that aren't proven with a placebo-controlled trial probably are going to end up not working. You know, I'll throw out a little, you know, if somebody says to me the other day, like, we're going to stop a trial because, you know, we're going to create a criteria that once we start a clinical trial, we will stop the trial if there's, you make a rule that says we'll stop a trial if there's less than, if when we look at the data, there's less than a 10%, there's less than a 5% chance that the drug will slow down ALS by 10%. And I think sort of the irony of that type of statement is that that's sort of, I think, our starting point on most treatments at best. So if you're taking experimental treatment, chance it's going to work, it's actually extremely low. And you know, I would much prefer that my patients sort of stick with the drugs that are on the market first. Like, don't throw out the things that we have good trials on and gamble on something that's just sort of out of the blue. I mean, that's the what's behind door number 3 mentality, which I get how people think about it. Hold on a second. My computer just turned off. 1 second. Hey, I'm back. Did you guys lose me for a second there?
No, we can still see you.
Oh, did you ever see that episode of The Odd Couple way back when Oscar had to type his name in really quick and he kept typing his name wrong because he had too much pressure on him? I just mistyped my password like 4 times because I was trying to get back in so quick because the computer kicked me off. But yeah, I'd much rather see my patients take drugs that we know work first. Like that, I think, is a good starting point. Like if you don't want to take Radhikava because you don't want a line, I get that 100%. But there's some drugs that work and we can deal with those in a clinical trial because roughly 50% of the patients can go into placebo and you can split up the treatments equally. But when you get into these random drugs, it just makes it very, very hard.
This plays perfectly into the next question. I know we speak quite often about the right to try. So the question becomes, why is the platform trial model not becoming the norm for studying ALS drugs? Why do you think we are still doing the old-fashioned trials rather than exclusively working with platform trials?
That's also a great question. I mean, a lot of that is just about, as good as the platform trial is, I think the platform trial is a great idea and it will grow. Like, I'll start off saying that. And it's early though, right? It's early days of the platform trial. They haven't had a success yet. You're a company, you have really good financing. You don't need the platform trial. You're going to hire a professional CRO and figure out if your drug works yourself, right? Like, it just at this point depends on who you are. On the other hand, you got a great idea for a drug. You're Joe from Tuckahoe, and you got some great ALS idea, and you got $9.23. We're all going to want the person to study that idea. So you're going to do anything you can to bring along. So there's a range there where the platform trial makes a lot of sense because it's efficient. You have a placebo group that you can divide up with other trials. So it's almost like human fast testing. It makes a lot of sense, but it doesn't make sense for everybody. Eventually, you get 30 years up the road, 10, hopefully 5 years up the road, and those guys are going to get more experience. They're going to get more experience across the whole platform. It's just going to get better and better, hopefully. And it'll carry, you get the same centers enrolling patients, but it's early days for that. So I think, success will bring more companies in. And, there's also great CRO, clinical research organizations out there who can work with big companies and create great trials too. So I don't think they're mutually exclusive.
Okay, awesome. So the next question we got was actually from a patient who lives in Canada, and she's trying to get into some US trials, and she's running into some difficulty, and she's just I guess not hearing back. So I was wondering if you had any input on that or how to go about doing that.
Well, now that I've heard rumors that America's just ignoring Canada lately altogether, so it's got nothing to do with ALS. No, I don't know. It depends who you're calling. Like, we definitely have worked with Canadian patients in our trials, usually from Vancouver, the western parts. But we have RC Seattle, Vancouver, and whatever's next to Chicago or Detroit, and so there should be no problem to it. And I don't understand. I mean, I will say sometimes in trials, you do worry about patients who are just going to have a hard time getting to your clinic. And, I don't want people getting on planes when it's such a giant struggle to travel, and maybe they underestimate how hard it's going to be. And we'll be very frank about that, but we definitely would work with people from Canada. There's no major issue that I'm aware of, but maybe at other centers there are that I don't know about.
This is more of a gene-focused question. Why aren't responders of trial drugs seeking out to have gene sequencing done to identify drugs that are gene-specific?
Well, there's a little of that going on, for sure, to look at genetic profiles of people and compare it with study responses. But it's really hard to do because, number one, there's not that many people. Once you just say we're getting into genetic disease, you're cutting the population down because most ALS is sporadic, right? So if you're going to start running trials on genetic diagnoses, other than C9, it gets very, very hard. And other markers, it's hard to base an early stage trial. So if you're going to have 200 patients in a trial, and you end up with a marker that's only there in a fifth of them, you only have 40 patients, right? So it gets hard to learn anything from 40 patients because then chance starts to be a big issue. So all this stuff is, people think about this, they look at it, but it's very hard like to grasp it and make it reality at this point in time. A lot of people like to ask questions about like, this is a good idea and that's a good idea. But sometimes the answer is, it is a good idea, we just can't do it, you know?
Yeah, that makes sense. So we want to be sensitive of your time. So I think we'll make this last one the last question. And the question that we're going to ask is, why are most ALS clinical trials only about 6 to 12 months long? And is there a way that we could make a longer trial that enrolls slowly progressing patients and specifically looks for improvement based on a few years, like 2 to 3 years long?
Well, yeah, that's an A+ question right there. I mean, those trials would be great. I mean, I think that's another thing that's like, you really have to think about it, because absolutely you want to split off slow progressors and study them for longer periods of time, which you almost by definition, you could all see that have to do, right? Like the trial's duration has to sort of parallel the rate of progression, or you have to have so many patients in the trial, right? But both of those things, think about it. What's the problem with both of those things? Either carrying your trial out longer or having to put more patients in trials so you could see the effect. They're costly. They're very expensive. So, you know, ALS gets a lot of drugs. One of the things that maybe we, you know, can be a little bit thankful for is it's, in a sense, it's the best disease to study. So a lot of companies are going to come to it because it progresses sort of at just the right rate on the average to study. Alzheimer's disease is very hard to study, as you all learned with what just happened with that drug. Very confusing, progresses slowly. Other diseases progress slowly. Some diseases progress too fast. So people want to come to this disease because that 6 months where you could see a change is a good period of time to invest into a drug. Like at the end of the day, what do they say? They say follow the money. A lot of this stuff is just like, how do you efficiently answer these questions so we cure the disease faster? That's like really what underlays a lot of this sort of thinking. Now, in the ideal world, I do think like we're going to sooner or later have to get at the question that you asked. We're going to have to get at that question. We're going to have to get at the questions of what cocktails do you use once we have a lot of therapies out there. We're going to have to get at cost questions once we have a lot of therapies out there, because it's going to get prohibitively expensive if you have to use 5 or 6 different drugs. It's going to be a lot of questions to answer, you know, and hopefully we'll get to a point where we don't have to try that hard because these drugs all work great and we don't have to think about it.
Thank you so much, Dr. Katz, for sharing your insight and your expertise. And hopefully I think we did get through most of the questions. If there are any, can I send them to you?
I actually think we got through almost all of them. We pretty much covered, as you're answering one question, it would answer another one.
Yeah,
so
true.
we were very, very good with that.
Yeah.
We do have one that just popped up. I'll ask you right now. Many of the trials that we see are targeting the TDP43 protein. What do you think of that approach?
I mean, you got to go in that direction, right? That's like a big clue staring everybody right in the face. There's this protein that gets aggregated in all our cells. So, you know, it's super important to do everything you can. That protein is like really important because it's like there, and you know that protein is sort of like the epiphany of everything. Like, how do you you know, how does it get there in the first place? How do you get rid of it? What's it doing? How is it getting from cell to cell? So it's a super important question, and trials you're starting to see ideas that are designed around either removing TDP-43, which may or may not ultimately be the answer to the question. It could be building there without being pathological. It could sort of be like, you know, when the garbage is getting put out at the end instead of actually the cause of the disease. So very hard, but it's obviously something that needs to be targeted.
So I think we just have that last one, James, in the spreadsheet, and then we can wrap it up.
I believe that was it, unless I'm not seeing the last one that you're seeing.
I think it was, what are your thoughts on RCH4?
Yeah,
That one I don't even know. I saw that one pop up. I
Oh.
was hoping somebody can tell me.
well, if you don't know that one, and I think AMX 0035 popped up 1 or 2 times. Are we familiar with that one?
Well, everybody's familiar with that one. So again, like there's a drug that they did a really good trial on. And to me, that's, you know, that's the best you can do from a reality standpoint, right? The 4th R, like that trial shows a 20-odd%, 23 or 24% slowdown in progression compared with placebo in a well-done clinical trial. Now, I imagine with the drug being in front of the FDA, you know, the FDA is struggling a little bit with the size. I don't know this, but they're probably struggling a little bit with the size of the trial because it was relatively small. And they're going to have to struggle with, do we get it out there? Or do we have a repeat trial? But, you know, that's good data and it's a good program. And, you know, once somebody puts out a clinical trial with the placebo group, you have to kind of say to yourself, that's what we got right now. Let's use it. It's a lot better than just guessing at something. And like, if there's one take-home message, it's, you know, that door number 3 phenomenon. You kind of have to ask yourself, do I want to take what I have or gamble on something I really don't know anything about? And that's sort of a lot of where my thinking goes with this stuff.
Excellent. Thank you again so much. I think we got them all.
All
Zoe,
right.
James?
All right. Well, you guys have been wonderful in a sense because I don't see any of you, but you seem great. The vibes are coming through my screen are great. I just wish all of you the best, and I hope we cure this thing for everybody.